Set them up within the two patients,
Okay.
and then we can compare between the two.
Okay.
Yeah, good.
You're just running the plugin on the subset of events from 165 versus
Okay.
166. You have to do it twice, run the plugin twice and get the same year.
Mm.
But then you would have this variable gene usage for a
Yeah.
specific patient.
Yeah.
And set them side by side and see the difference.
Okay.
Okay, and we're only interested, if I'm taking it, between memory naive and plasma.
Right, that'd be wonderful.
And then we wanted to look at the variable genes.
Mm.
And so I think we're basically there in terms of what we were thinking that you might want to get out of this.
That's 160. What is that? Okay.
This is 165. So yeah, it might actually be good to label this, right?
Right. So there's a lot of plasma cells, and I think 166 is going to have less purple. That's one that tops out as a purple one. I mean, it's
Right, and
sort of
giving
interesting.
you memory in terms of your top variable gene usage,
Yeah.
which might just be might be the fact that you have very few memory cells. Yeah, compared with like naive, for example.
Yeah.
Okay. So now I'm going to, if this is all that we want to get for this comparison,
Right. For now.
okay, then I'm going to close out of this. Now you can always recreate this, right, by going back to the BJ Explorer, but you'll have to set back up this comparison. So then the next thing that Tim is suggesting, we run this plugin on the other sample, and that way we can compare.
Okay. So it's not like Teasney, you know, where you have like a different. I mean, I know there's no seeding point, you know, with
Yeah.
a static, you know, data sets with like the VCR. Yeah, no, I scratched that. Never mind.
Then, as Tim mentioned to begin with, I should have stuck with the same color scheme. I didn't, but let's just make sure I do going forward. So yeah, memory green, naive blue, plasma purple. And then if I explore the repertoire, we should be able to look at the variable genes associated with that. And actually, I see a similar pattern. I'm not sure that what's on the axis is exactly the same. No, it's not. Actually, not sure how it's arranging that exactly.
Can you save an image of that?
Right,
What's used.
in most use specifically. But then, I don't know why we don't get just a stacked bar chart that goes from high to low.
I think this, at least the left side has the memory.
Right. Let's study. This would be worthwhile studying. Of course, we want to be able to do it ourselves, and for other samples. But
Oops. And I also noticed I neglected to show my legend here, so I'm going to recreate that.
now this one here is who is this? 166.
This is 166.
Well, I think there's a few. There's more that end up not purple.
Right,
There's some that start out and they don't continue
right.
to plasma cells. I see naive, but these are not truncated. There's 165.
Keep in mind, these are only the rows that got mapped, or sorry, cells that got mapped to your
VCR.
ERN information.
Yeah.
So if you want to know like how many cells, memory naive or plasma, that's just going back to our original gating.
Hmm.
So you can see memory, there's only 62 memory cells in 166.
See, what would be interesting is to find one. Now, this gets back to Binga's question a while back. Is to get the actual numerical data for the blue versus the purple here on this, and then see which one seems like they survive. Which plasma cell, B cell, has a greater ratio of purple to blue here. I mean, I can kind of get it with my eye. I can see some lot kind of low blue and big purple, but right, like that one, IGHV 3.7. Is this who is this?
It's 166.
Is 160.
166.
So, and some get truncated, like this one, IGH 459.
Really biased.
Right. Yeah, there's some bias here.
Right,
This is exactly what I was thinking.
right.
This is 165
66.
or 66. Okay. So you can see you can see the bias. Some start out low, but they're all. Look at that one. That's all plasma cell. There's almost no naive, but there's a lot of plasma. And others, like some of those, but others start out with some naive naive and a lot of plasma versus, you know what I mean? The purple to blue ratio. Can you show? So this is 166.
166.
Is
So
there?
this is 166, and we have a static image for 165, which is here.
Right.
I was also thinking we could put these side by side.
Right.
Right. And you saved a picture of this anyway, right? A copy of the
Yeah.
image.
These pictures are all contained in the labs.
I don't have to go back to my camera. This is 165.
This is 165. That's right.
Oops. Yeah, this is 165. There's 166.
Yeah, what'd be nice is to have this data some other way and then export it. Oh, yeah, is that right? Yeah, can you export this? Because this would go right into an Excel spreadsheet or something. Export, yes. That's
This
it.
one I'm not so confident in. Let's see, save graph data.
See.
Try and set it to my downloads.
I guess it's a CSV.
Yeah, it'll be a CSV.
But that's
My
okay.
question is whether or not it will accurately contain it. Data was no, not writing a repertoire chart out.
Really? We want this thing.
Yeah, I'm not sure. Try one more time.
Yeah, I don't think it's gonna save.
Why not?
No, didn't work. Well, take note of that as well. That's just a bug in the current implementation.
Is the data anywhere other than on this image chart? Is actually the ratios somewhere? I mean, it must be somewhere.
Right, but it's not accessible as far as I can tell. The exact, you're looking for the exact number of plasma with this gene.
Yes, right.
I mean, if we knew, okay, one way we could kind of work around this is if we know that, in this case, remember we found the most common variable gene was 33-3.
Yeah.
Uh, we could now ask how many are plasma and how many are naive by just sorting on these populations.
I see. How do we do that?
And this was already beginning. 3-3 was, and that should appear here. That's way too many. That can't be right. Let's try this for the plasma though.
This is a lot. This is a lot of potential. The other one can't do it. I don't think B-loop can do
No,
this.
I think they both, this one cannot see through showing the sequence.
Right, but the other one, but it could put things together.
Right. So
We're just saying, Ian, this is, we were just over there talking to the guy, David Crossman. I don't know if you remember him, but he's the local person who's doing our analysis, and he uses VLoop. And VLoop can do things that this can't do, but this can do things that VLoop can't do, such as what you're doing right now. So, you guys,
Well,
you
we
need to
would
get
be really interested to learn the things that you guys want to do that we currently can't do. So some things that I wrote down is, Circo's plots
Right.
dig deeper into these subpopulations, even within the comparison, because I know we set up originally naive, memory, and plasma, and we want to then look at what are the different patients within that.
Yeah. Right.
Big
I
ask.
mean, what you're doing right now, I think
We like it.
this is what a biologist would want, right? They want to know what is the negative selection or positive selection? What is the survival of an autoreactive plasma cell? What determines that? What's the underlying mechanism? What's the transcriptome of these cells that survive, and we'll figure out if they're autoreactive by making the antibody. But if they are autoreactive and they shouldn't be there, then you look at the transcriptome of those cells. But the first step is to see, is there even a population to study? But it looks like there is. So that's actually, it's a kind of it's not even a new question. It's like, I remember it came up 30 years ago, but we just were never able to answer it. And now you're so close. You're right there. You just get this. Tell us why the ratio of plasma cells to naive are higher in certain ones. By looking at the transcriptome underneath, right?
Right, that's right.
Did they express IL-21 receptor or what? Why are they there? They shouldn't be there. This is the solution to autoimmunity. Ian, you solved the problem. It's been
Well,
around.
that's your job.
Ladies and gentlemen, I'm sorry I have to jump off the call to be on.
Okay.
Thanks for joining, Tom.
Really, thank you, Ian. I'm learning with you guys from Ian today. So thank you for your time.
Thank you.
Thank you.
Thanks for coming, Tim.
You're welcome. Take care.
Thanks, Ian. All right. So that's it. You're so close, Ian.
Well, what I can do is let me clean up this naive B cells that have this IgHV 333 phenotype. I think this is wrong. But besides that, other things I was thinking I wanted to get into was like, now that we have, say, clonotypes for plasma B cells, we could compare this between samples 165 and 166.
Yes, that would be wonderful.
Right.
Okay, you want to try that now?
Yes.
Yeah.
Yeah, we get out of here. I think we got all the images we wanted for that.
One
Before we go
thing.
and crash this, let me save it.
One thing, Ian, since this is such a burning question, we actually have sequences. I just found out they finally got around to sequencing naive and plasmablast.
Transition.
Transitional. Yes, even earlier. And wasn't it plasmablast? We have the sequence for those for two patients, two subjects. So we sorted, and so we won't have to have hazy gating on. Maybe we don't know
That
what.
would allow us to compare this, go back to.
Oh, right, yeah, we can take this and go back to that, because that's actually our naive and plasma. I mean, from surface markers that are accepted. This is kind of a guessing by the gene, at the gene level, which are naive, or which are naive in plasma. But, we can compare the new data, we have it on two subjects, with what we get here, and see if we're really identifying plasmablasts properly, and sort of it'll become confirmatory. So I mean, this is the big question. This is it. And then, what is the difference between those plasmablasts? Or is that what you're going to do now? What's the gene?
What I was thinking to do is come back to this QC population, and then go back to the gene view,
Yeah.
and look at within each of the patients the different plasma.
Exactly. Yes, let's do that. That would be wonderful. What's driving the plasmablast in 165 versus 166?
What did I choose here, sorry?
This is another 30-year-old question.
So this would be 665, and over here I'll choose 66.
Well, people have this gene, that gene, but they don't know. They look at gene by gene.
Yes.
This is it. It's
40,
not-
not 30.
All right, 40-year-old question. Maybe even longer. It goes back to 1909 with that hypothesis, horror autotoxicus hypothesis. But nobody knows. Gotta get this. the big moment. Yeah.
Right.
And then same thing for, this will be the inverse of that, so it's gonna be up in 165.
They have to work.
Secret.
And we know that this is actually versus the plasma B cells in 166.
Isn't that amazing? Wow.
Secret reveal.
We
Wow.
get these two gene sets. And there we go.
What do we got?
Oh, I think it's a similar one. We kind of
So
know.
this is up in 165, so the disease, more disease, right? And then this is up in 166.
Are you going to send these gene sets to us?
It's in this file.
Better yet, I mean, I'm just going to keep saving this analysis, and I'll send you the geek zip, and then all of this stuff should be saved in there.
It's interesting because I see some interferon genes over here.
Right. But same.
'Cause
I
we
find
actually-
it, same,
And
same one. We saw.
really?
I think these are the genes that's constantly upregulated.
Right.
Throughout different stages in 165.
Oh, right.
Very
'cause-
trying.
Right.
Sample is erroneous, but
Is there a way to sort of plot these or look at the, what do you call it, the Disney plot of,
yeah. So I haven't calculated t-SNE for this data set, but I do have UMAP, and so we can look at it in UMAP. I was actually curious to look just at the different subsets we identified in UMAP, so let's check that out. So UMAP is just an alternative to dimensionality reduction compared to t-SNE. And I might have wanted to rerun this because we got very discrete populations here. A little bit worried that, not sure what exactly happened there, but try and center them up a little bit more.